I can kick it off with one question to George regarding the example you used on the flames as, and use a cell, use a chemoheterotroph as an example of the surprising capacity of life to just burn organics and then survive. But that's not the complete system, right? And there's a second part to the system of a photoautotroph that is sequestering, which is just as remarkable. Neither of those systems by themselves would explain the phenomena that interests you.
Well, I mean, if you want a really global view of things, what makes it all work is that the sun spits out photons at 10,000 degrees. A single 10,000 degree photon ends up being a whole bunch of 4-degree photons, and the second law is happy. So all the rest is just technical detail.
Trying to connect all three talks. So many people think of schizophrenia as a critical transition and bifurcation. And so how can we use Cas9 to show that and show the theory behind? Is there a path towards that?
So can we ask you to say, you said many people see schizophrenia as a what? I didn't know. A bifurcation, critical state transition and things like that. So what would be the level of black boxing we could address and to use that framework? So
how would you split up consciousness into pieces that were simple, tractable boxes that connected rather than a series of genes?
So you could, so on the highest level, one could think about cognition, hallucinations, delusions as large as boxes that were, that you might understand individually, that together, if you, if your cognitive box is abnormal, you proceed up a certain path, but at the stage at which your hallucination box is encountered on that path, if you have certain things in your hallucination box, you bifurcate and you can't actually go back. So I think that's the kind of thing you might think about. Now, you've also asked the question about reducing that then to the molecular level. So let me see if I can go the next route, which would be if we get to the stage where we understand enough about human brain circuitry, both at the gross and molecular level, one might begin to connect circuits to specific, to the specific underlying boxes. And if we could put our circuits in the boxes, then we could ask individuals with their fabulous Cas9 technology to perturb the boxes, perturb what's in the boxes, and ask whether we bifurcate differently, or can you, if you have the cognitive box and you get to the hallucination box, but you've decreased things, the box is less full, does that allow you to proceed and not bifurcate in the same way? So
I guess since this is a technology symposium, I'll address the technologies part of it, which is, I talked a lot about sequence manipulation. There are SNPs in the genome that you can introduce, but there are also maybe even ways to try to make that even more reductionist, right? There might be a lot of different ways to achieve different levels of expression. And Stanley Chi, Jonathan Weissman, Wendell Lim, and the various especially have come up with ways to inactivate Cas9 and then change expression. And the thing they're working at, so basically you attach transcriptional activators or repressors to Cas9, catalytically inactive Cas9, and you increase or decrease expression, right? The same way that you're seeing small changes in multiple genes. So they're currently working on multiplexing that, and they're also working on allelic series. So the idea being, rather than turn everything all the way off or turn it all the way on, you get tiny little changes. And then if you could multiplex that hugely, you might have some kinds of technologies and tools to ask the complex questions, right? To build up the more complex changes that might lead to the more
Right. And
interesting phenotypes.
So I think you've, from my perspective, you've hit the nail on the head around what the problem is in the way sort of the traditional way biology and neurobiology think about approaching this. I'm fairly certain that all of the changes are going to be subtle and small, and there are probably many of them, and they'll have to be, maybe we'll get lucky and there'll be something that's large and you find some node and you make a magic bullet drug and, and everything is great. But the reality looks to be much more complex. And so how do you model that? It doesn't fit easily into knocking a gene in and knocking it out and asking, in a rodent whether it, performed in the Morris water maze better than its, littermate. So these kinds of clever genome-wide, system-wide strategies are critical from my perspective. So.
Bill, you had a question?
So on schizophrenia, the
Yeah.
results, I guess, earlier this year of C4 complement potentially in neural pruning, making it into a interaction of developmental networks and potentially with an immunological component to it. Any expansion on that with respect to your work?
Not yet, right? Obviously, I think everybody is working feverishly to place those results in context. I think that most likely it will be one of multiple pathways that are involved. The work is very nice and very, very lovely, but there are, it doesn't account fully for the genetic signal in that region. And, they've chosen sort of a candidate phenotype, which again is persuasive, but one perhaps among many. So I think it's going to take some time to know and figure out what C4 is doing. It's also an extraordinarily complex locus to investigate.
Anushree?
Okay, Dr. Corin, I found the research on CRISPR and Cas9 especially fascinating. And while I recognize that it's a method mostly predominantly for preemptive measures for genetic disorders, do you have any propositions for using the same sort of genetic editing for a maybe fully developed human body to prevent disorders, or possibly for testing a cross-species genetic editing, in testing it out in mice or something?
Well, I guess I would answer that in two ways. Number one, very certainly gene editing is not just for genetic disease. I think that the way that I actually got started on this was in a research mode while I was still at Genentech, right? The first papers showing Cas9 as RNA directed just changed everything, because we were working in a very basic biology system. I work on ubiquitin and deubiquitinases. That's sort of the biology I'm interested in. And to me, genome editing is a way to answer questions that we just never had tools before, right? And a lot of the questions you might ask are totally relevant for human health. So we used it to ask questions about signaling pathways that we couldn't answer before. We used it to make models that we could never make before. We made them incredibly quickly. So from that point of view, gene editing can also be used to just take traditional therapeutic discovery, target ID, all that kind of, and just turn the knob to 11, right? You can suddenly do things so much more quickly. The other thing that I would say that I'm really enthusiastic about is just the power of gene editing for totally fundamental research. Non-model organisms especially. There are people at Berkeley, there's one guy who's been doing, Nipan Patel, who's been doing editing in non-model crustaceans and butterflies and just things you would normally never look at. And you think to yourself, well, what possible use could that be? Think about everything that we know about human health and the ways networks go together are in large part put together by analogy to other systems, to model systems. But when you look under the lamppost, you build your examples based on what you've seen before. When you start to look at all these other systems, you start to discover, things don't necessarily work the way they do in the top 3 models that we've looked at. I think we're just going to learn more and more, and it's, in some ways, that's the most explosive growth of gene editing for me, because those are the places where we'll have totally unpredictable breakthroughs, right? We won't know what's out there until we go looking for it. Who knows if this crustacean has some gene that controls limb regeneration that we just, it's not present in some of the models we've looked at. now we figure out the way that these things work. So I think for me, that's one of the ways in which just total undirected scientific curiosity and the enabling power of Cas9 can lead to big changes in human health.
Looking forward to it. Thank you.
Just an interesting tidbit here. The question was asked by a high school junior, which is remarkable. ISP is about education. It's great to see with Emerging Technologies you have emerging talent also here to absorb the new information. So thanks for the answer. I had one question regarding the resilience and robustness concepts. I think you skipped over resilience in interest of time. And you mentioned that biological systems can live in highly variable environments. And I don't know if that is true. I mean, it's probably true that they live in changing environments, but they're spatiotemporally defined. And take organisms that are photosynthetic, they oscillate between daytime and nighttime physiology. So that gets to robustness. But I think the resilience aspect, which is to me more interesting, which is unique to biological systems in many ways, is the ability to recover from stress. And I guess the critical slowing down kind of gets at that, the ability to withstand increasing amounts of stress and yet recover up to a point that you can no longer recover. So do you think that's a distinguishing characteristics of biological systems versus other?
I don't know if it's distinguishing, but also we have to be careful not to confuse life as we know it now with life as it was originally produced. But if you take an organism like, Deinococcus radiolaris, it will survive at 120 or some incredible temperature. It dehydrates in high radiation fields, and it stops tap dancing when you freeze it. But it comes back alive again. And the bacilli, you can make spores which last for thousands of years, and many systems are very robust. And we tend to think about misreading our error rate in making proteins as being just not worth the effort cleaning up that last bit. But it could be that last bit is actually what keeps us going in times when things get unexpected. So the mathematicians who work on these kinds of things have shown that if you have A to B going by a series of steps along the way, but there are many ways that you can get from A to B, that those systems tend to be much more able to recover from perturbations than systems that have straight lines.
Let me ask one more question regarding Cas9. So Craig Venter just published a paper on the reduced genome. I always struggle to figure out what to make of it. It's a great technological achievement, but you coming from the other side of removing genes, and they coming from the other side of synthesizing genomes. Do you see a way of combining the two to, you mentioned designer babies yesterday, but more biosynthetic pathways for renewable chemicals and such? Would they benefit by combining the two?
Oh, absolutely. So there are several groups at Berkeley and elsewhere throughout the U.S. and the world that are trying to do large-scale pathway engineering using Cas9. It's more, again, it gets to the non-model organism idea, right? If you have, let's say you have some synthetic chassis, right? And you work in that synthetic chassis because it's genetically tractable. Well, maybe there's a different chassis that's better for bioproduction. It's just not genetically tractable. Well, maybe now it's genetically tractable.
So how many systems are being engineered? Do you have a rough guess?
You mean how many places have people tried Cas9? Or
how many types of organisms have been subject to CRISPR-Cas9 in a successful way?
So I actually stopped keeping track. I had a list for a while, and I stopped keeping track because it just got...
What was the last count?
I think I stopped at like 40 or 50.
450?
What?
Are there notable failures?
Well, it's hard to predict. It's hard to say that because most people don't write papers saying, we tried to edit this organism. No, I know that there are certain organisms where people have trouble getting things to work. So, for example, yeast, Cerevisiae is a great example because Cerevisiae is great for homologous recombination, but people are having trouble doing knockouts using Cas9 because they don't like to do NHEJ. But then there are related yeasts that will do NHEJ and don't do HR. And so it's sort of a matter of not like the enzyme doesn't work. It's a matter of, again, everything comes down to repair pathway choice. And so to me, it's more about the enzyme is doing the cutting, and now we get to learn a lot about the ways that different organisms repair their genomes and take care of damage.
Can I ask a question about this sort of detailed processing? It is clear that this is an exceedingly important task if you want to ask very specific questions about very specific proteins or whatever. But the question is, how far is that going to carry you? Does biology, by building highly interacting nonlinear systems, take the perturbations from one system and smudge them out to the point where you basically can't get useful information very quickly? Or is it going to be more like, the story that we're hearing now in cancer, that there actually are occasional cases in which a single point makes an enormous difference in how boxes interact, if I can put it that way?
So I guess, I should make it clear, I'm not a bottom-up person, I'm a top-down person. So I'm rather than trying to build the thing synthetically, I'm trying to understand sort of how things evolved. So one of the ways that I got into using genome editing was by doing design of protein dynamics. The idea being that, we think about proteins interacting with each other, and that's really important for function, but the speed at which they move may be just as important. And so we made biophysically characterized motions, characterized them by NMR, and then replaced the endogenous genes such that they still had all the reaction partners, but they just moved at different rates, and then try to figure out how that affected phenotype of the organism, sort of invisible state, and how that regulates fitness. So I think from that point of view, it's all how you use the tool, right? Cas9 is just a tool, and you can use it to ask questions exactly like you just put out there. You can go top down, you can go bottom up, and it's just a matter of what you do.
But I know that I don't need to do string theory to play Beethoven.
Yeah.
Right. And so there is such a thing as you can say this isn't important, and the question is how far do you have to go? I don't know the answer to that. Bill?
So a question on gene editing. Imagine perturbations of this nature becoming so cheap that one can do a whole genome perturbation base by base to get the functionality of each base, and then do it at 2 base pairs, 4 base, 10 base pairs, and get a multi-scale kind of what's the function of this stretch? Is this totally pie in the sky, or is perturbation become so cheap that that's doable?
I think it's just a matter of time.
Okay.
It's just a matter of time. At this point, several labs, ours included, have started to make huge libraries. So we have this really fantastic collaboration with Agilent, where they have technologies to print arrays of oligos, very high quality. We've used them to tile across entire topologically associated domains, megabases at a time. We're not trying to replace individual bits, but there's say 10 genes in there, but most of it is energetic space. How does that energetic space regulate the transcription of all the genes in that surrounding area? So we're not doing this genome-wide yet, but we're trying to take apart this sort of stuff for certain areas that we think are going to be interesting. Yep.
So my question is to George. So you kind of hypothesize that there is like a critical transition in evolution of life when we had, let's say, critical amount of number of reactions that were put together so as to give rise to something that is alive. So in the context of, let's say, what Craig Venter's work shows, that there are, like, let's say, around 400 genes that are necessary, or minimal essential number of genes for sustaining life. So do you think that this transition happened only once and this is what we see? Or do you think that it happened several times but none of them survived and this is what we see?
Really hard to judge. And remember, what Craig's work is really, really beautiful, but when you think about a protein, it's probably complicated to say that you just need 400 isolated things. You need 400 pieces, or whatever the number that is involved there, which have the right network of connections. And the right network of connections is now a much larger number than the 400. So all of these issues of how much information there is in a given string of bits, it gets a little bit confused in biology. I mean, after all, the genome itself is not giving you information about the protein, it's giving you the shipping label for a protein that has evolved to solve a function. So it's the name, it's like Alice. It's not, this is a protein, if you do this kind of thing, it's a protein that takes HMG-CoA reductase and serves that function. So I don't know the answer. And also remember that particular organism has the characteristic that it requires a rich environment, so that all the amino acids are being made by somebody else. I'm not sure what a minimum requirement is.
Actually, if there is no other question that I'm going to ask you, Jacob. So I'm kind of curious and wondering why nobody asked you this before. But I mean, CRISPR-Cas9 still has a lot of off-target effects, right? So what is the cutting edge right now, and what are the efforts to reduce those off-target effects?
So To me, I think it sort of depends what you want to do, right? Obviously, we're interested in therapeutics. And when we think about therapeutics, off-target effects are a really big deal, especially for things like hematopoietic editing, because you want to edit 10 to the 7 cells and you can't clone them out, and you get really scared about being able to even see off-target effects when you're going to give someone a therapy that they're going to carry for the rest of their life. If you're thinking about this from just biological discovery, hypothesis generation, or gene editing for modeling, very often what I tell people is, it's no real different from siRNA or shRNA type experiments. You just want to complement your phenotype. You want to have multiple clones. It's nothing different from that respect. So in that case, you don't necessarily need some sort of incredible, off-target free engine. You just need to know where your off-targets are and be able to show that your phenotype comes not from an off-target, but from the real thing. On the therapeutic side of things, there are a bunch of technologies that are out there. Very recently, people have described mutated Cas9s for different off-target properties. They have reduced off-targets. We've played around with those, and they do have reduced off-targets, but they also have reduced overall activity. So they're about a third to a half as active as wild-type Cas9, but the off-targets fall off faster than the normal activity. So, depending on what your off-targets are, you may be willing to live with that, you may not. The other thing I would say is that depending on the locus, people have described guide RNAs for which there are 0 off-targets, and people have described other guide RNAs for which there are hundreds of off-targets. And we don't understand why exactly some have a lot of off-targets and some do not. And so that's one of the things that we're trying to look at in large scale.
So this is a question for all of you. Essentially, we saw a very nice example how editing can help us to cure a disease, or how important are individual SNPs and NQTLs for another human disease. But on the other hand, we saw George demonstrate that a very, very simple system can have 2 different phenotypes. And essentially, if we were sampling only few seconds after the initiation or 1 minute after, we could see something completely different. And this is more or less what we are doing in human health right now. I mean, we can just go as someone is going to the hospital or not and can have one certain phenotype or not. We miss totally what can be the different states in physiology or disease. So my question is, once we have this library with all these different editing, all these different positions, and how we are gonna scale up to understand phenotypes, or in what scale we need to look the phenotypes? You think always it should be the human as a disease, or in completely different scales?
Can I just make a remark about that? There's a very interesting sort of system that if you're interested in that question, you might look at, which is learn enough about artificial neural networks to see how they work. Because the characteristic of the neural networks is that you can take a neural network and you teach it your telephone number or whatever it is, and you take a pair of scissors and you cut it in half, and it still remembers your telephone number. And the reason is that we have a model in which function or information is stored in bins. It's the sort of computer model. But there's another model in which information is dissociated over a net. And they're very different in the way you're instructed. So that, for example, if you looked at this recent business with the Go playing program, AlphaGo. AlphaGo absorbed 50 million Go games and did whatever it did. And it really is good. But if you ask AlphaGo what it does, it can't tell you, and you can never understand. So it really is different. You have to ask totally different questions when information is delocalized in a way that you cannot associate a particular function with a particular structure. And I think it's very interesting in what you do and very interesting in how the mind works, because you're, in a sense, seeing that there's no single sharp point of defect. There's something else going on, but we don't really know what that something else is right now.
Right, and I would suggest that it's possible, this may be heresy, but it may not be that we require any kind of one-to-one correspondence with what we would traditionally call phenotype in order to improve human health and human mental illness, right? So being able to work out that detailed molecular some kind of very specific molecular mechanism that may in fact just not be necessary. So we don't know that yet. Right?
Okay, I think that's about the time we have. So thank our panelists again.